Alright, in this video we're going to go through some revision notes for Biology 0610, Cambridge IGCSE for Topic 4, Biological Organic Molecules and Biochemical Food Tests. So let's begin. Biological organic molecules are all molecules with hydrogen atoms and carbon atoms strongly bonded to other carbon atoms. So you have hydrogen atoms and carbon atoms, both strongly bonded to other carbon atoms. It's like hydrogen and carbon bonded to a chain of carbon atoms. Why do organisms need biological organic molecules? Two reasons. The first one is to provide energy for life processes and provide raw materials for growth and tissue repair. There are four main molecules in living organisms: carbohydrates, fats or lipids, proteins, and nucleic acid. The first one, carbohydrates. The chemical elements present in carbohydrates are carbon, oxygen, and hydrogen. Second, protein. Chemical elements present in protein is carbon, oxygen, hydrogen, nitrogen, and sometimes sulfur. Third is lipid. Chemical elements present in lipid is carbon, oxygen, and hydrogen. Now let's go through each one in detail. The first one is carbohydrates. Carbohydrates are long chains of simple sugars. There are three types of carbohydrates: monosaccharide, disaccharide, and polysaccharide. Monosaccharide is only one simple sugar, for example, glucose. Disaccharide is two simple sugars, example, maltose. And polysaccharide is lots of glucose or lots of monosaccharides mixed together, like starch or glycogen or cellulose. Mono means one, so one simple sugar. Di means two, so two simple sugars. And poly means many, so lots of glucose together. Large molecules are formed by smaller ones. For example, when we take the example for the formation of polysaccharides, we have many monosaccharides, so many small molecules that combine together to form a large molecule like polysaccharides. This is an example of carbohydrates in organisms. For example, in liver cells, you have glycogen granules, and you have cellulose in plant cells for the cellulose cell wall. The next organic molecule is lipids. Lipids or fats, both are the same thing. Lipid is a molecule formed by condensation of triglycerides. Triglyceride is one glycerol chemically bonded to three fatty acid chains. So if you see the diagram here, we have one glycerol chemically bonded to three fatty acid chains. 1, 2, 3. So one glycerol chemically bonded to three fatty acid chains is a triglyceride. Lipids can vary in size and structure. They are solid fats at room temperature and oils in the form of liquid at room temperature. The third organic molecule is protein. Proteins are long chains of 20 different amino acids joined together in a particular sequence coded by genes. Example of protein is keratin. Keratin is the protein present in hair and nails. These are some facts about proteins. Amino acids can be arranged in any order. Amino acids are the basic building blocks of proteins. They make up proteins and they can be arranged in any order. All protein have the same basic structure, but the R group for each one is different. Function of protein depends on their shape. So each protein molecule has a different function depending on its shape. Amino acids are soluble in water, hence easily transported in living organisms to take part of cytoplasm reactions in the cell. This is the general structure of amino acids. So they all have the same structure, only the R group is different for each one. How are proteins formed? This is a simple illustration. So we have amino acids, then amino acids join together in a chain to form peptide, and then these chains fold together to form a protein molecule. All right. The last organic molecule is nucleic acid. Nucleic acid is two strands of DNA in a double helix structure. Double helix structure is the twisting structure of the DNA. Something like this. This is known as the double helix structure. Okay, DNA. DNA is deoxyribonucleic acid. It is a molecule with instructions of growth plus development for all organisms. And DNA consists of nucleotides, individual DNA units. Nucleotides are divided into three main groups. The first one is 4 nitrogen base in pairs, adenine with thymine, and cytosine with guanine. These 4 nitrogen bases in pairs are held by the hydrogen bond. So the hydrogen bond holds these 4 nitrogen bases together. Then we have sugar and phosphate. These form the backbone of the DNA strand. So let's have a look at the diagram below. Here we have adenine with thymine, cytosine with guanine, 4 nitrogen base pairs held together by the hydrogen bond. And then we have phosphate and sugar on the outside to form the backbone of the double helix structure. And with that, we are done with biological molecules. Now we are going to enter the next part of this chapter, which is biochemistry. It's not really biochemistry, it's just an introduction to biochemical food tests. But before we jump to biochemical food tests, we just need to understand the definition of biochemistry and some related facts. Biochemistry is the study of organic and inorganic molecules, inorganic like water and ions, organic as we took above. Metabolism are chemical reactions in living organisms. This is the simplest definition for this chapter. We're going to study metabolism in detail in chapter 7. Hydrolysis is a reaction that splits subunits that make up large molecules. For example, for the formation of triglycerides, it's done by hydrolysis and condensation. Formation and decomposition of triglycerides. This is an example of hydrolysis where large molecules are broken down to smaller ones by the addition of water. So we have starch. So this is a plant. Maize, it contains starch. After adding water or H2O to this plant, what happens is that hydrolysis occurs and large units are broken down into smaller glucose units, which are later broken down into glycogen for the rat. Condensation is the opposite, where large molecules are built up from smaller ones by the removal of water. So hydrolysis, you add water to break down molecules. Condensation, you remove water to build up molecules. All right. Now is the introduction to biochemical food tests. We have about more than 4 biochemical food tests in our syllabus. The first one is test for glucose reducing sugar test. We're going to take the steps for each test, and usually the questions come around for 4 to 6 marks. So you must memorize all the steps and always include these last 2 steps in all experiments in paper 4 and paper 6. Take safety precautions with goggles and heat-proof gloves. Repeat the experiment for validity. I advise you not to use validity and accuracy because most examiners do not accept accuracy. So use validity. Accuracy is optional, but not advised. Alright, so let's begin. Test for glucose reducing sugar. Step 1. Add Benedict's solution inside the test tube for the sugar sample. Step 2. Heat or boil the mixture in a thermostatic water bathtub for 5 minutes at 60 to 70 degrees Celsius. Step 3. After you have finished boiling the test tube, take the test tube outside of the thermostatic water bathtub and observe the color change. If it's positive, it's going to change from blue to orange to brick red. This is a positive result. If it's negative, the color will remain as it is. It is going to remain blue. The last 2 steps, take safety precautions with goggles and heat-proof gloves. Repeat experiment for validity. Accuracy is optional. The last 2 steps is a must. For all experiments. So let's see what's going on here. This is an illustration for this test. We have a solution to be tested. This is the sugar sample solution. We add Benedict's solution to this food sample, and then we heat this for 5 minutes at 60 to 70 degrees Celsius in a thermostatic water bathtub. This is the thermostatic water bathtub. And after that, we observe the color change. Again, safety precautions is a must. Safety goggles, heat-proof gloves, test tube with tongs. This one is optional. Rules before performing a Benedict's test. It should always be performed on equal volumes of unknown solutions. So you have let's say 2 unknown solutions, or for example you have you want to compare the sugar solution of A with sugar solution of B. Okay, both should be on equal volumes of solution, and it should also be on equal volumes of Benedict's solution. So we have, for example, 2 test tubes. We're testing Benedict's, or we're testing reducing sugar test in 2 test tubes. The solution volume should be the same. So if it's 5 centimeter here, it should be 5 centimeter here. And the Biuret solution volume should also be the same. So if it's 2 centimeter here, it should be 2 centimeter here. Both must be equal. All mixtures are to be heated at the same temperature for the same time. If this one is heated for 5 minutes, this one should also be heated for 5 minutes. If this one is heated at 30 degrees Celsius, this one should also be heated at 30 degrees Celsius. So same solution volume, same Benedict solution volume, same time of heating, and same temperature of heating. Alright. The next one is test for protein. Step 1. Add drops of Biuret solution to the food sample, not Benedict's, Biuret solution, and then observe the color change. If it's positive, it's going to change from blue to violet purple. If it's negative, the color will remain as it is, it will remain blue. Again, the last two is a must. Take safety precaution with goggles and heat-proof gloves. Repeat the experiment for validity. This is the test for protein. We have the food sample here, and we have the biorate solution here. As we add the biorate solution, if it is a positive result, it will change to violet purple. And if it's a negative result, it will remain as it is, blue. What are some hazards of biorate solution? There are two hazards. The first one, it contains copper sulfate. It's dangerous for eyes, so we should wear safety goggles. Next, it contains sodium hydroxide. Sodium hydroxide, it's corrosive for the skin, so we should immediately wash hands. Sodium hydroxide, the formula of sodium hydroxide is NaOH. This is just for extra information. All right. The third food test is test for lipids or fats. Step 1, add 2 centimeter cube of ethanol alcohol to the food sample and gently shake it. Step 2, pour the mixture into a test tube containing equal volume of distilled cold water. Note that wherever you see an underline, this means that this is the main part the examiner is looking at in your sentence. So in all these steps, you must include the underlined part because this is the part that's going to get you the mark. Step 3, observe the change. So after you put it into the tube containing equal volume of distilled water, if it's positive, it's going to be a milky white or / cloudy emulsion. And if it's negative, the liquid will remain clear. Again, last two is a must. Take safety precautions with goggles and heat-proof gloves. Repeat experiment for validity. 1 mark for each. So we have sample of 95% ethanol, should never be 100%, and distilled water. After performing the experiment, if it is a positive result, it will form a milky white or cloudy emulsion. If it is negative, it will remain clear. A quick fact about lipids: they are insoluble, hence they cannot make aqueous solution of fats and oils to biochemical tests. The third biochemical food test is test for vitamin C. Step 1, add 1 centimeter cube of DCPIP solution to the test tube. Step 2, add small amount of food sample as a solution. So in the previous experiments, we have a test tube, we have the food sample, and we're adding the solution. In this case, we have a test tube. We first add 1 centimeter cube of DCPIP solution, and then we add the food sample. After we have done this, we observe the color change. If it is positive, the blue dye will disappear. If it is negative, the color will remain blue. Again, last two is a must. Take safety precaution with goggles and heat-proof gloves. Repeat the experiment for validity. Here we have DCPIB solution. We have the sample to be added. If it's positive, and if it's negative, it's going to remain blue. And if it is positive, the blue dye will disappear. Vitamin C solution strength. Comparing the solution strength with its drops, it is inversely proportional. If you have few drops, then you have strong vitamin C solution. If there are more drops, then we have weak vitamin C solution. Okay. Now, test for starch or carbohydrates. First, we add drops of iodine solution to the food sample and gently shake it. So we have a food sample, we add drops of iodine solution. Then we observe the color change. If it is positive, it will change from orange, yellow, brownish color to blue-black. If it is negative, the color will remain orange or yellow or brown. Of course, last two, take safety precautions with goggles and heat-proof gloves and repeat the experiment for validity. Here we're doing a test of starch on a potato. So we first peel the potato, then we add iodine solution, gently move the potato and observe the color change. Since starch is present, that's why it changed to blue-black. Otherwise, the color would remain orange or yellow, but here it's brown, so the color would remain brown. Okay, hazards of iodine solution. It is irritant to the eyes and causes allergy, so we should wear safety goggles. And with that, we are done with the chapter. So we have covered biological organic molecules, the four types of biological organic molecules: fats, proteins, carbohydrates, and nucleic acid. And after that, we have covered the food tests for proteins, reducing sugars, starch, vitamin C, and so on. So that is all for this chapter. Now we have some summary tables to summarize everything. This table shows the substance and whether it uses heat or Biuret or Benedict's and what is its positive and negative result. If it is present, we put a tick. If it's not correct, we put an X. This is a past paper style question. So first one, the substance is lipid. Does lipid use heat? No. Does lipid use Biuret? No. Does lipid use Benedict solution? No. Does lipid have a positive result blue-black? No. Does lipid have a negative result milky emulsion? Yes. Next, protein. Does protein use heat? No. Does protein use Biuret solution? Yes. Okay, so we're done with protein. Starch. Does starch use heat? No. Does starch use Biuret? No. Does starch use Benedict? No. Does starch have a positive result blue-black? Yes, because when we add iodine solution, when we observe the color change, if it's positive, it will be blue-black. Last is glucose. Does glucose use heat? Yes, glucose is the only one that uses heat. Molecules and their reagents used, their positive results and their negative results. Protein, the reagent used is Biuret reagent. Positive result is violet purple. Negative is blue solution. Lipids, reagent used is ethanol alcohol. Positive result is milky solution. Negative result is clear liquid. Starch, reagent used iodine. Positive result blue-black. Negative result yellow solution. Glucose, reagent used positive result orange brick red, negative result blue solution.